fatal pathway with dopaminergic neurons originating in the substantia nigra. So when we consider the remaining neurons, we can observe abnormal inclusions called the Lewy bodies, and these inclusions are mainly composed of aggregated alpha-synuclein. So here is the patient. So I think the video, there is an issue with the video. So she has tremor, rigidity, bradykinesia.
Video in your presentation, it's not possible.
Okay, no problem. So, and this patient had abnormal posturing, as you can see, with flexed posture. And this patient had also a sister with the same disease. So if we consider the UKPD Society Brain Bank criteria for Parkinson's disease, there is an exclusion criteria. In fact, having more than one affected relative was considered as an exclusion criteria. And these criteria were established in 1992, and they are still widely used. In fact, Parkinson's disease for a long time has been thought of as the archetypal non-genetic disorder. Till 1996, where the first gene for Parkinson's disease has been mapped, and it at that date, a large Italian family with an autosomal dominant early onset pathologically confirmed PD was reported, and genome scan and linkage analysis was performed in this family. And the locus on chromosome 4, referred to as part1, was associated with PD in this family. And soon thereafter, mutations in the alpha-synuclein gene were identified in this Italian kindred and also in other families. And in this Italian pedigree, there was a mutation, a substitution mutation, causing the disease in this family in the alpha-synuclein gene. So the alpha-synuclein protein is involved in signal transduction, membrane vesicle trafficking, and cytoskeletal dynamics. And when we have mutations in the gene, in the alpha-synuclein gene, it leads to the death of the dopaminergic neurons and to the misfolding of alpha-synuclein with abnormal aggregation. And as we've seen, there are these abnormal aggregates of alpha-synuclein are the main component of the Lewy body, which is the hallmark, the pathologic hallmark of the disease. So there are several mutations reported, such as the substitution we've seen in the Italian kindred, but there are also copy number variation and mainly duplications causing the disease. So this was the first monogenic form reported of Parkinson's disease, but since then, we turned out that monogenic forms of PD represents up to 10% of PD cases. So to date, we have 23 loci identified, 19 genes involved in PD with either autosomal dominant, autosomal recessive, or X-linked mode of inheritance. And the most frequent one is the PARK8, or related to mutation in the LRRK2 gene. It's an autosomal dominant with a late onset PD. So, and this locus for PARK8 was mapped in 202 in a large Japanese family. And mutations causing this PARK8 linked Parkinson's disease was identified 2 years later, and it was in the LRRK2 gene. And the LRRK2 protein is a very large multi-domain protein with kinase activity and also GTPase activity. So when we have mutations in this gene, it leads to the death of dopaminergic neurons due to the influence of this mutation on various cellular pathways, especially on mitochondrial function, on the trafficking of vesicles, on microtubule dynamics. And also LRRK2 can lead either directly or indirectly to the accumulation of alpha-synuclein and of tau protein. The most frequent mutation is the glycine to serine substitution at position 2019, and this mutation is the most frequent one, and it causes a gain of function with an increase in the kinase activity of the protein. So here we've studied a large number of familial cases of PD from Tunisia, 92 patients with familial PD, and we've performed comprehensive sequencing of the LAC2 gene in these patients. So we found that up to 39% of our familial cases has this founder mutation, the G2019S mutation in the exon 41. And then we've looked for this mutation in our apparently sporadic cases. And we were very surprised to find that up to 30% of our apparently sporadic cases had also this same mutation, this founder mutation. And as you see here, the study made the cover of the Lancet Neurology because of its important conceptual implication, as it brought the familial and sporadic PD etiology closer than they have ever been. So in fact, this mutation, the G2019S, is frequent in Tunisia, but also in the other North African countries, and representing about 30% of sporadic cases and 40% of familial cases, and also in Ashkenazi Jews. It's much more rarer in the other parts of the world, representing only 1 to 2% of PD cases. And there is a founding LAC2 haplotype shared by all these patients, and the common founder of the mutation dates to about 2,600 years ago. So the question is LAC2 PD and idiopathic PD the same disease, or are they different diseases? To answer this question, we've studied the motor phenotype of our LAC2 patients as compared with the idiopathic PD patients. And as you can see, the phenotype was quite similar. So the median age of onset was of 58 years in our heterozygous patients versus 58 years in our idiopathic PD patient. So it's a late onset PD. And the tremor was, in most cases, the first symptom. The PD phenotype was mainly mixed in both groups. And we've also compared, most patients were heterozygous, but some of them were homozygous. And we've compared the motor phenotype between heterozygous and homozygous, and there were no difference in severity. So the mutation does not seem to have a dose effect. And then we've also followed our patients prospectively over 6 years, and we found that the motor aggravation, the motor progression of the disease was quite similar in LAC2 patient and in idiopathic PD. So I can't, unfortunately, show the videos, but we have a very dramatic improvement in levodopa in our LAC2 patients. The same response we can also observe in our idiopathic PD patients. But these motor symptoms are just the tip of the iceberg. In fact, Parkinson's disease is a multi-system disorder with the cognitive, psychiatric, sleep dysautonomic disorders. So we've assessed these non-motor symptoms in our LAC2 patients, and we've assessed the autonomic function. We found similar results in both groups. Also, olfaction dysfunction is the same in both groups. The only difference which was significant is regarding sleep disorder, and mainly REM sleep disorder, which was less frequent in our LAC2 PD patients. We've also assessed cognitive function in our patients, and there were no difference in cognitive dysfunction if we compare GD2019S carriers versus non-carriers. So overall, the motor phenotype and non-motor phenotype was quite similar. We can distinguish clinically between LAC2 and idiopathic PD. And it's an autosomal dominant disorder, but with incomplete penetrance, which is age-dependent. So for instance, here we visited a family, and the patient had the age of onset of Parkinson in this patient was of 45 years old, and his mother, who had the mutation, was 101 years old, and she had no Parkinsonism. So that we have a variability in the age of onset even within the same family. And if we consider the patients aged below 50, so the penetrance is below 20%, and for patients over 70 years old, the penetrance is over 80%. So how to explain this variability? It would be due to either genetic factors, genetic modifiers, or environmental factors. So here we've performed a genome-wide linkage and association study in our patients, and we found that a single SNP in the dynamine 3 gene was associated with the age of onset in our LAG2 patients. So when the median onset of LAG2PD in GG carriers was 12.5 years younger than that of CC carriers. And also the age of onset was found to be related to environmental factors, especially smoking. So smoking was associated with a later median age of onset. But of course, I will not advise our patients to smoke because of the other issues related to smoking, but it's protective in these patients and it delays the onset of the disease. And very recently, we've identified a new gene involved in autosomal dominant PD. It's RAP32. And through a linkage association and functional analysis in 4 Tunisian patients, and this variant seems to increase the activity of LAG2. So we've seen 2 autosomal dominant forms of PD. The first one, which is rather rare, and the second one, which is the most frequent cause of monogenic PD worldwide, especially in North Africa. And now we see 2 other the common causes of autosomal recessive PD, which are part2 and part6, PINK1 and PARKIN. And there are a large number of mutations in causing the disease in both these genes, ranging from substitutions to whole exons deletions. So PARKIN is an ubiquitin ligase, and PINK1 is a mitochondrial kinase, and both of them interact and are involved in mitophagy and in the degradation of the damaged mitochondria. So when we have mutations in these genes, we have mitochondrial damage and oxidative stress. And we've studied 231 familial PD patients belonging to 90 Tunisian families, and we performed direct sequencing and dosage analysis of PARKIN and PINK1. And as you can see, these mutations are related to an early age of onset. So here in PINK1, the median age of onset was of 35 years, and in PARKIN, it was of 32 years. So it's much younger than the age of onset observed in either idiopathic or LAG2, which was here of 60 years old. So these autosomal recessive forms of PD are causes an early onset PD. And the PARK mutation frequency was of about 4% versus as high as 50% of the familial early onset PD in Western studies. So PARKIN-related PD is much less frequent in our country. PINK1 mutation frequency was of 18% versus only 1 to 2% of early onset PD in Western studies. So in Tunisia and also in North Africa, we have a much higher prevalence of PINK1 compared to Western countries, to Caucasian patients. And so we found 4 substitution mutations in the PINK1 gene. And the motor phenotype, the clinical phenotype, was quite similar to that, except of these earlier age of onset. It was quite similar to idiopathic PD and to LAG2 PD, with the good response to levodopa, with the tremor as a first symptom, etc. And the only difference was a much more frequent dyskinesia, which are normal movement related to the levodopa treatment. So far we've seen the monogenic forms of the disease where we have a rare mutation with an important effect, highly penetrant mutations. But in most cases, in the idiopathic PD, which is a complex disorder, we have an intrication of both genetic and environmental factors, and we can have variants, genetic variants, which are associated with the disease, and these variants are much common, are frequent, but with a much lower effects. So to look for these variants, we need to perform very large association studies and to compare PD patients with healthy controls, and we need for that bioinformatics, and we need new techniques of sequencing, next generation sequencing techniques. Because of the very large number of patients and controls we need to study, as the effect is really small. So here several PD risk factors have been identified, mainly in the genes involved in the monogenic forms of the disease, such as the synuclein gene or the LAC2 gene, but also in other genes, the GBA, glucocerebrosidase gene, and the MAPT gene. So, and the most frequent risk factor for PD is the glucocerebrosidase mutations, especially in Jews. But we've looked for them, and we, in our population, these mutation does not seem to be a common risk factor for PD. We've also looked for synuclein and MAPT variants in our PD patients, and the MAPT variants were not associated with a susceptibility to PD in our patients, but several variants in synuclein seem to increase risk of for sporadic PD. And here is a very large study, META-GWA study, where they studied 7.8 million SNPs in 37,700 PD patients, and they've compared them to 1.4 million controls. So through these meta-analysis, they've identified 90 independent genome-wide significant variants associated with PD, and they could define a polygenic risk score based on these variants to determine the risk to develop PD for a specific individual. But the issue is that these studies were performed mainly in Caucasian populations, so this risk score is not appropriate for our patients. And we need to perform our own study, as these variants are ethnic specific. So to sum up, we have some rare mutation with the highly penetrant effects, such as the mutations in PARK1, PINK1, synuclein, uncommon mutations with the incomplete penetrance, such as LAC2 mutation, and much more common variants, which increases the risk of PD. So why is it important to determine these genetic factors involved in PD? In fact, it helps us to understand better understanding of the pathophysiology of the disease, and also to develop precision medicine. So, and LAC2 is a very crucial target for neuroprotective therapy, especially in our country where it's very prevalent. So far, the treatment of PD is mainly symptomatic. So there is a deficiency in dopamine. We give levodopa to the patient, substitution therapy. It doesn't stop the progression of the disease. There is so far no neuroprotective therapy for Parkinson. So targeting LAC2 would be neuroprotective drug. And as we've seen, the mutations in the LAC2 gene are causing a gain of function and increase in kinase activation. So, and more recently, a study showed that even in patients with idiopathic PD, not only the patients with LAC2 PD, in idiopathic PD, they've observed an increase in the LAC2 activity. So how to target this LAC2? It could be through LAC2 silencing or through pharmacological strategies. And currently there is a an ongoing phase 1 study with the antisense oligonucleotides to silence the LRRK2 in early onset PD in patients with the onset of PD under 7 years. It's still ongoing and in the phase 1. And there is also another strategy which is using LRRK2 kinase inhibitors. There are several inhibitors which are available. They're with high specificity toward LRRK2. They are potent. They cross the barrier, and they seem to be safe in prolonged use. And there are several clinical trials with these LRRK2 inhibitors. Ongoing study, and the one is we have a phase 2b study, the LUMA study, which is a multicenter randomized double-blind placebo-controlled study in early PD patients who had developed with the diagnosis below 2 years. And so this is oral treatment with LRRK2 inhibitors, and it's still ongoing. So there are a lot of challenges with these treatments regarding safety, regarding biomarkers, regarding efficacy, when to treat the patients. It could be too late when we have the clinical symptoms. So when we should start this neuroprotective therapy, a lot of challenges to be tackled. So thank you for your attention. I would like to 20 minutes. So thank you for your attention. I would like to take this opportunity and to invite you to join the MDS, the International Parkinson and Movement Disorder Society. So there are lots of membership benefits, and if you are interested in movement disorder, it's for free for us for Africans. So it's an opportunity to take. Thank you again.
For the movement spirit to Pakistan, no assistance to provide to two questions from the time. So on two questions to send.
Amira? Excuse me. Thank you.
Thank you very much, Sami, for this very nice presentation, and we see that similar interests for Parkinson ALS, but studying the genetics, of course, is very interesting because these slides therapeutic development.
Yes.
So the questions regarding whether there are some redundant biomarkers that can be used for follow-up of patients during treatment.
So biomarker, there are mutation. It's a kind of biomarker, yeah. But the problem with the mutation is that so there are biological biomarkers and also the mutation. The problem with the mutation, to take it as the only biomarker, is that, as we've seen, the penetrance is incomplete. So we have a patient which is 101 years and she has no Parkinsonism. So we need to have other biomarkers if we consider starting the treatment. So if we have the mutation, even if you have the neuroprotective treatment is available now, it would be useless to use it in such patients. So we need to have additional biomarkers for the disease. So sorry? Neurofilaments are associated, not specific to LRRK2, it's related to degeneration in general and axonal loss in Parkinson in general. It's not specific for LRRK2. So we have clinical biomarkers such as olfaction tests to look for these genomic symptoms which are early in the disease progression, such as constipation, psychiatric symptoms further, clinical biomarkers such as depression, constipation, olfactory loss, and also REM sleep disorder, which is early. So we can rely on these clinical biomarkers. And there are also biological biomarkers, but the problem they're not really validated in LRRK2. So they are we can dose the activated substrate of LRRK2 in the urine and in the serum, but not really validated. till now. And the problem is also there are new biomarkers used in Parkinson's disease with the alpha-synuclein amplification assay. So it's validated in geographic PD, in CSF. But the problem is that in LAG2, the neuropathology is diverse, so it's not always alpha-synucleinopathy. So in half patients we have alpha-synucleinopathy, in half patients, in the other half, we have either other inclusions like tau inclusions, TTP 3843, or even no inclusions. So we need to validate the alpha-synuclein amplification assay in our patients because of this high prevalence of LAG2 PD.
Any more questions? To Dr. Samir?
Is there any tourism?
Email.
Is there any, first of all, thank you very much for your excellent presentation. I would like to ask if there is any genetic applications to treat patients with Parkinson's disease, such as correction of certain mutations, for example.
So there is it's ongoing, but it's phase 1. As you've seen here, we have trials with the antisense oligonucleotides to silence LAG2. Also in GBA there is also trials in PD related to GBA mutations. But they are ongoing study and early clinical study, phase 1. And there is a phase 2B trial, the LUMA study with the LAG2 inhibitor, because the mutation causes in LAG2 PD an increase in the activity, so we can use an inhibitor. There's been a phase 3 study, Lighthouse study, but unfortunately it has been stopped. We were selected as our institute was selected as a center to take part of this phase 3 study with LAG2 inhibitor, but unfortunately the study has been stopped.
Thank you, Dr.
Hello everyone. Now we will move on the next presentation with Professor Amira Metwali. Professor Amira Metwali was one of the graduates from the first class of pharmacy biotechnology at German University in Cairo. Right after graduation from the German University for Cairo, she decided to pursue her postgraduate studies at Germany, starting with Master at University of Heidelberg and German Cancer Research Center, followed by PhD at Technical University of Munich in the field of nutrition and immunology, and resulted in a not regular degree but a PhD with distance. Currently, Dr. Amira works as a lecturer and postdoctoral researcher, chair of nutrition and immunology at Technical University of Munich. Welcome, Dr. Amira. The floor is yours.
Thank you. Do you have a pointer? Pointer.
All right, until we organize the pointer, I think I can start. Thank you very much for the introduction and thank you for the invitation. I would like to particularly thank Dr. Kaitz and Dr. Osama and Dr. Mohammed Hamid for the invitation to be here today, first time in Tunisia and first time to contribute to the German Arab Network. It's my pleasure to be here, and my talk will be about microbiome signatures and host microbe interactions, and how we can use functional genomics to unlock therapeutic potential and to identify biomarkers. So first of all, I'd like to give you a little introduction about gut microbiome in particular, because that's where I spent the last 10 years of my research. And so, that's okay. I think I can use yeah. Perfect. Oh, yes. All right. So just a little introduction about gut microbiome. So there has been great interest in gut microbiome in the last 2 decades, in particular, so in the last 20 years, which coincided with a high advance in high throughput technologies, high sequencing technologies, and the generation of genomics data. And this was reflected here in the number of publications. So here we retrieved all publications which addressed microbiome and its association to different diseases over the past 20 years. And you can see there is an exponential increase in the number of publications over the past 20 years, reflecting the interest in gut microbiome. And here you can see that there has been a lot of research, and we have identified a lot of connection or evidence that gut microbiome is associated with a lot of different diseases, namely inflammatory metabolic diseases, but also neurological diseases such as Alzheimer's, and also asthma, for example, and arthritis, and of course cancer. So colorectal cancer, there is evidence that colorectal cancer is highly correlated with changes in the gut microbiome. All right, so moving forward, here what we wanted to do in this meta-analysis, that was an effort we did at Technical University Munich, where we summarized basically data from microbiome studies across the globe, and we focused mainly on 2 disease entities, so inflammatory bowel disease and type 2 diabetes, and we wanted to understand why is it challenging to identify microbiome signature, or that can be used as a biomarker basically in clinical settings. And so here what you can see is basically a summary. So we summarized based on the meta-analysis all the bacterial taxa which are either enriched or decreased in type 2 diabetes or IBD in different population studies or patient cohort studies. And what was very, I wouldn't say surprising, so it was expected, but what was also very evident from the meta-analysis was that there is a high heterogeneity in gut microbiome data. So the inter-individual variations between populations, between patients or just persons, healthy persons, was quite high, which is a challenge to identify a consistent biomarker. And by a biomarker here, I mean specific configuration of microbiome's composition or structure that is specific to a certain disease or pathological condition, for example. And so that was our first conclusion, that there is a high heterogeneity. The second conclusion was that we just looked at 2 disease entities, IBD and type 2 diabetes. And within these 2 entities, we could see that there is a huge overlap in the bacterial taxa which are associated with both diseases. So yes, you can see here in the Venn diagram that there are some taxa which are either increased or decreased, for example, in IBD in particular, and some which are increased in type 2 diabetes. But we also have a huge overlap here that which makes it very challenging to have a specific biomarker that we can use to basically predict response to treatment or predict whether a patient or whether this person has IBD, for example, or type 2 diabetes. Yeah, so that, I mean, that and a lot of other conclusions we've, sorry, I keep going. We have summarized here in this Nature Reviews, Gastroenterology and Hepatology, which was published recently. So if you're interested, you can go and look it up. And we also, based on the meta-analysis, proposed a number of recommendations on how you should plan a microbiome study if you are planning to identify microbiome signatures. So again, that was also a definition which we proposed in the review, that microbiome signature means a unique pattern of microbiome configuration that can stratify defined physiological and pathological condition and can be used to predict the risk of disease, development or progression, or even response to treatment. And that was another effort we did in a frame of an EU Horizon 2020 grant, which basically combined around 30 partners from different
Excuse me.
Oh, yeah.
Yes.
All right. Okay. All right. So again, that was an effort we did together with around 30 partners in the frame of an EU Horizon 2020 grant. And in this grant, we had different partners from key experts in microbiome research in Europe. And the aim of this grant, with multiple work packages, was to identify challenges and to propose recommendations in the field of microbiome research, to move the field of microbiome research forward, basically. And so anyway, we had in this one work package, the aim was to identify state-of-the-art future or definition of microbiome-based biomarkers, and to provide recommendations on basically the challenges and how we can improve biomarker discovery for microbiome-based biomarkers. Yeah. So basically, I wouldn't go through each recommendation here, but I would advise you to go to this review and have a look. And basically, again, what we identified as the main challenges were the high inter-individual variations, high variability, the lack of standards, that people use different standards and different protocols for the generation and for the analysis of microbiome data that generates a lot of heterogeneity, for example. And also the lack of curated databases. And that's why I was really intrigued by Dr. Kai's talk this morning, because I think we need curated, well-annotated databases where people would basically deposit their data sets, and then we can use it to advance the field. All right, moving on. Yes, so in my research, I focus on two main entities, IBD, inflammatory bowel disease, and type 2 diabetes. However, I spent the last 10 years focusing on inflammatory bowel disease. That's why I would like to give you an overview of some projects where we used multi-omics integration to identify biomarkers for IBD. And just as a quick overview, so IBD is an inflammatory bowel disease. It's a chronic relapsing condition, and it comprises several subtypes, but mainly two main subtypes. These are Crohn's disease and ulcerative colitis. Crohn's disease is mainly focused in affects the small intestine, and it has a patchy manner. Ulcerative colitis is mainly in the colon, and it can extend to the rectum as well. And here you can see the phenotype of Crohn's disease and ulcerative colitis. So it's a very nasty disease, and it's chronic, and it keeps coming back. So and so far there is no treatment for IBD, and all the treatments are basically to manage the conditions, and some patients remain to be refractory to all available treatments. So here again, that's so multi-omics integration provides a very good tool to understand and to study complex diseases such as IBD. All right, and here that's a typical pipeline of how you can employ multi-omics integration for the study of IBD. So basically, you start by having a well-defined cohorts of patients, and for these patients, as you heard earlier in the talks earlier, metadata is very important. So you need to understand as much as you can about the patients, their genetic background, their diets, whether they get any kind of treatments, whether they have received antibiotics. For example, since we look at microbiome, so these are all important information we need to have. And then we collect samples from the patients. And for IBD, we focus on fecal samples, stool samples, or also mucosa-associated bacteria, so basically biopsies from inflamed tissue. So that could be small intestine or colon. And then we apply different layers of data. So genomics, metabolomics, shotgun metagenomics, targeted metabolomics, for example, and we use multi-omics integration and sophisticated machine learning models. And then eventually, to bring this to the clinical side, we need to validate the biomarker, the candidate biomarker, using in vivo or in vitro models as well. Right. All right, so basically I will just show you one case study, and that was published in Nature Communications 2020, where we looked at a cohort of Crohn's disease patients, and these patients had refractory Crohn's disease, so they didn't respond to any conventional treatment. And the only resort then was to treat them with hematopoietic stem cell transplantation, and that's not a routine treatment, it's just reserved for this group of refractory patients, right? And so we followed these patients over time. So we collected samples at inclusion and over time, every 6 months for 5 years. And it was extensive sampling. And of course, we did it in collaboration with our partners in the clinic. So that was in Barcelona. And we had then eventually stool samples, biopsies, and blood samples collected from these patients over time. What we did was we started by looking at the microbiome composition, and basically here what you can see is a multidimensional scaling plot. It's like a PCA plot, if you wish, And what you can see, these dots represent microbial community of one patient at a specific time point. And we followed, for example, this one patient over time. So when they started the treatment, and then over time when they went in remission and at some point relapsed. And what you can see here is that the patients go through a lot of fluctuations in their gut microbiota composition during the course of disease, and that these changes correlate with the disease activity. Okay, and so what we did next was, again, looking at try to separate between patients who are in remission or those who are in relapse, and to see if we can if we can separate these patients based on their gut microbiome profiles. And indeed, we could see a separation between the patients, and that was reflected as well in the community richness and diversity. So we saw that patients have much lower diversity, microbial community diversity compared to patients during remission. And so based on the taxonomic composition only, we used a 10-fold cross-validation classifier to see if we can classify the cohort into diseased and non-diseased based on the information of the microbial community structure. And what you can see here that we had an area under the curve of 0.79, which is not great. If you talk to clinicians, you can use calprotectin, which is an ELISA, a simple ELISA, and this ELISA would give you a prediction of the patient inflammation stage with more than 80% accuracy. So why would you bother to do 16S shotgun metagenomics and do all of that to reach exactly the same thing that you can do with the 16S? And so here we I think I'm gonna jump into the functional potential. So basically, when we did the 16S analysis, that wasn't enough, obviously, to classify the patients. So that's why we moved on to do metabolomics. So we did untargeted metabolomics on the 4 cohorts. And you can see here in this volcano plot that we identified some features which are significantly enriched in disease or in non-disease conditions. However, that was not significant. And when we looked at and the functional potential of microbial communities, we identified some pathways which are involved in sulfur metabolism. So that gave us the idea that maybe sulfur metabolism or sulfate-reducing bacteria could be specific to the inflammation in IBD, or at least in this cohort. And that's why we then decided to move on to the in vivo validation. So what we did here, we established a humanized mouse model, or a human microbiota-associated mouse model. And this mouse model is basically, as you can see here, these are germ-free mice, and germ-free mice are born and raised in sterile conditions, so they don't have any microbiome, and that's why you can use them as models or as mini incubators that you can modulate. You can add which bacteria you're interested to study, and then follow and understand microhost interactions in this model. So we had a model of IL-10 knockout, that's a colitis mouse model, and it was germ-free. And at Technical University Munich, we have one of the biggest germ-free mouse facilities in Europe. So we established this mouse model by transplanting fecal samples or gut microbiota from patients at different disease states and different conditions into the germ-free mouse. And what was very nice and very interesting is that the mice reflected exactly the phenotype of the patients. So you can see here a histopathology score, and you can see that mice which received microbiome from inflamed patients got inflamed, and those who received microbiome from patients in remission did not get inflamed at all, and they have a histopathology score of 0. So that was reproducible for 10 different microbiomes from 5 different patients. So that was really great, and we used it then as a model to study further. And basically, we replicated what we did in the human cohort, but now in the mouse, to see if we can replicate the same signature which we identified in the human cohort, but it was a little bit diffused due to the heterogeneity in the mouse model. And what you can see here is, again, so here you can have the human donor, and then each circle here combines all the mice which are transplanted with gut microbiome from this one particular donor. And you can see that each group of mice always reflects the gut microbiome composition from the respective donor, but also you can see that there is different configurations which can lead to the same phenotype, basically. So we have different microbial communities which can be correlated to disease, but they are completely different, and the same goes for these which are disease-free, which tells us that taxonomy doesn't tell us much. It's more about the function, because you can have different taxonomic groups, but they do eventually they infer functions which lead to disease, right? All right, so basically, we also looked at the metabolomic signature and using metabolomics and shotgun metagenomics to look at the functions, and we confirmed that we indeed see enrichment of sulfated compounds, mainly bile acids, so tauroconjugated bile acids, and also metabolic pathways involved in sulfur metabolism. And eventually, when we integrated all the data now, from human and mouse, and including the metabolomics, shotgun, and 16S data, we could see that the classifier now has a much higher predictive power. And I keep doing this. So you can see that now the same classifier we used earlier and gave us a 79% predictability is now improved to give us around 96% when we integrated all data sets. So multi-omics integration improved significantly the predictability of disease outcome. And based on these clusters of OTUs and metabolites, we identified a set of 7 OTUs or 7 bacterial taxa, which are relevant to disease in this cohort, and we identified eventually the sulfovibrio, which is a sulfate-reducing bacteria, to be enriched in disease. So eventually, this is my conclusion. So basically, we identified, we saw that IBD patients have a very heterogeneous microbiome signature. We saw that humanized mice reflected the disease phenotype of the patients, and that the integration of multiple omics could improve the predictive power of disease predictability. And eventually, we summarized our proposition about gut microbiome signature and how we can use it to improve biomarker discovery in this Nature review again. And you can see here basically how we can implement that. So I get the signal that I need to rush. So I think you can read it in your free time. But thank you very much for your attention. I just want to acknowledge my group at Technical University Munich and all our collaborators and all our funding bodies. And thank you all for your attention.
Thank you, Dr. Amina, for this nice presentation. If you have any questions, you have a few minutes. After the break.
No, just Céline Mahmoud now, online, the president.
Yes or no? I'm sure.
Hi.
Okay, thanks, Dr. Amira, for the interesting talk. Just starting question regarding the microbiome studies in general. So, from my experience also in microbiome research, I find that mainly in studies usually goes for the time series data in order to study the dynamics of the microbiome activities across certain time or certain time span. So from your experience, what would be the shortest time span you have that can be, let's say, accepted by the community for microbiome studies? I have seen 2 months, 2 years. Some studies also go for 3 years. So what do you think about that?
Yeah, I think it's a great question. I think it depends on the disease you're looking at. because it always depends on the dynamics of the disease and the disease scenario. So if you look at a cancer patient, you can expect that you need to have a long-term study. If you're looking at acute inflammations, probably you don't need that. Or if you're looking at, for example, infants, and you want to see the progress after birth and then introducing formula or solid food, then probably you don't need also that. It also depends on for example, if the patients take antibiotics. So general recommendation, for example, is to exclude any patient who received antibiotics for the last 3 months, right? That's not always possible when you look at different diseases. I mean, if you look, you cannot design a study. Primary response we look at is around 6 months, and then we look at remission again at 1 year, and then we look again at 2 years to see if they have maintained remission. So that's an approximation for IBD, for example. And IBD, you have a significant correlation between inflammation and microbiome signature. It's not the case for type 2 diabetes. It's variable, yeah.
One short question
Yeah.
regarding the metabolic that you have actually.
Yeah.
How do you make sure that the metabolites that you are making, are microbiome or
Human related,
Exactly.
yeah, yes, of
How
course.
do you make it that?
I mean, of course, I mean, you, at the level of when you do the annotation of the metabolites, then when you look at the ontologies, then we exclude all human-related metabolites if they exist on the databases, right? And metabolomics are not really well annotated, as you know, so more than 50% of the metabolites are not annotated. So you cannot completely exclude it. And it also needs the experience of a chemist who's looking at your mass spec and who will tell you that that's probably a microbe-generated metabolites or a human-generated metabolites. Yeah, a bit tricky. Thank you.
Thank you very much, Professor Very nice presentation. So I think that we have a question that we have time to discuss. So I have an announcement to do. So we are running out of time. So for the workshop, will be parallel. So for those who will follow the workshop, so they can go now. And for the rest, you'll be here to continue with the presentation. Okay, so now we can continue with our presentations.
Now we will continue with the next presentation with Dr. Cyril Mahmoud, who will talk about genome screen advanced biotechnic solutions for modern bioinformatics. The floor is yours, Cyril.
Thank you so much, everyone. I will just try to share my screen if you don't mind, and then if it's better.
Cyril?
Yes.
Are you hearing us?
Yes, we do. Yes, I hear you.
Hi, Cyril.
I am hearing you. Do you hear me?
Cyril?
Yes, I hear you. Do you hear me? Can you hear me? Par contre نسمع فيكم مليح. Alors je vais essayer de.
ثانية. هو البروجرام تاعها محلول الهواء النووي خرج الصوت من البيسي مش خرج الصوت ما نعرفش.
Probably you hear me now. Do you hear me now? Do you hear me now? I'm not sure if it's working then. Hello, so it is working and also speaker, headset, earphone.
Do you hear me?
Yes, I do.
Yes, I do.
But with a lot of echo, so I'm not sure if you can fix that.
Yeah, we have to close the mic.
I mean, probably what we can do, if you are hearing me, we can also start and muting yourself from your side. But if there is any question afterward, we can do it. I mean, let me know if you approve this solution.
It's done.
Okay, cool. So I can, okay, you can start. Great. I will just start, share my screen if you don't mind, so that, because I have made some modification. I'm sorry, I cannot hear you, but feel free to stop me or unmute yourself. So first of all, thanks a lot for the invitation, Dr. Osama Khamasi. I really appreciate this invitation, and it's my pleasure because I had made actually some training within Institut Pasteur probably 11 years ago. So that's a pleasure for me to have a small presentation within the amphitheatre of Institut Pasteur Tunis. So for all other people coming outside Tunisia, welcome to my country. So making a bit of introduction of myself. So I'm Dr. Celine McNean. I am team leader within GenScript. So I'm team leader of European countries and also Africa. So I'm taking care of project evaluation, discussion with the several institutions, also Institut Pasteur Paris, but also a lot of other universities, pharma in Egypt and also in many other African countries. Well, so of course, the presentation will be about mainly the advanced biotech solutions for modern bioinformatics. Of course, this is very important for me to know with whom you are dealing with. Just a brief introduction about GenScript, of course. So GenScript is CRO, basically. Okay, so it's has been built, I would say, 21 years ago. It was founded in New Jersey, in US, in 2002. We have begun first with gene synthesis, but now I will show you that we can do a lot. We have several entities just to know our geographic attendance. We are in US, UK, we are in Netherlands, in China, and also Singapore, Japan, and also Korea. So the idea of GenScript is also helping researchers, pharma companies, research institutions, and also startups to kind of mature the projects from the research and development phase till the preclinical phase. So just to have an overview again of what we can propose for your research in general. I mean, basically, to be honest, you can think about this in anything you can do in your lab. So we can cover the part of molecular biology, so everything related to gene synthesis, plasmid preparation, cloning, subcloning, everything related to gene editing. That means CRISPR-Cas, guided RNA, but also HDR. We can do also protein expression actually in several expression system, whatever. Sorry. So it could be mammalian expression system, could be bacterial or insect. Our golden star, honestly, is our turbo. Probably I need to make the pointer. That's better, so that you can see with me. Yeah. So turbo antibody expression system, which is able to produce the antibody starting from only 5 business days. So if you are also working a lot within the mRNA, so we can also do the IVT mRNA template. It's not here in the presentation, but certainly you have heard about the 2 new trends, I would say, for the mRNA, which is the circular RNA and also the self-amplifying RNA. We can also help in oligos, peptides, cyclic peptide, neoantigen peptide, and so on and so forth, virus cell lines, and also antibody generation. So if we don't have the sequence, we need to immunize certain animals. We can also do that, plus the downstream application. So that was very important for me to know with whom you are dealing. So GenScript is really very well known, even with the big top 5 pharma all over the world. I have chosen, though, 2 services, which, because when I have discussed with Dr. Usama Khammasi, I just wanted to make this synergy between what GenScript can do in terms of biotech versus the bioinformatics and also the AI. And the most, I would say, innovative part, which is now accelerating the research, is, of course, the artificial intelligence. So it's not new to everyone, of course. Every now we can just have also an answer within few seconds. And now we want to have, between brackets, a protein optimized, an antigen optimized within few minutes. So that's why we wanted to be as fast as the follow, or I would say the flow or the rhythm of the bioinformatics and also the AI. So of course the cycle is usual. We are designing. Could be a protein, could be peptide, could be an antibody. We build, we test, and then we learn. So for sure, we need to be very, very fast, because with the machine learning, with the database we need to nurture each day, we need to be fast. So that's why I wanted to, I mean, it's not new, but the platform now is available for even pharma and also research institution. And the idea is about, of course, the CFPS, so the cell-free protein synthesis. We can make several proteins now in 5 business days only. Why? Because AI startups or also bioinformatics startups or research institutions wants to have this quick protein expression in order to be able to analyze quick, to nurture quickly the machine, the algorithm, and then produce this machine learning algorithm for the optimization of, could be enzyme, could be proteins, could be antibody, and so on and so forth. So of course, you know that cell-free expression system, you are not using any living cells. It's just some, I would say, amino acids, energy buffers, some DNA. We mix it all with the cellular machinery. We can produce this protein within 5 business days. So of course, for those 3 parts, for protein expression, traditional bacteria, mammalian expression cells, I would like you to focus maybe in the second or the middle one. So here we can produce a protein in less than 1 week. Honestly, which is really very important, especially with people who are working with the CAR T cells or doing any gene editing or gene therapy or cell therapy. That's very important. So we can basically produce the VHH and also the SCVF, for example, within only 5 business days. Well, why? Okay, we can ask why we can just go for the cell-free expression service. It's very fast. We can do then more than 1,000 samples per day, and it's very cost effective. I would say probably it's $120, something like that, by protein, which is really, really nothing. So apart from that, just make a bit, I mean, I would not go to too much into details. I think this day has been very, I would say, tough of details. Probably now we are getting into the end of the day. So what we do for the CFPIs is very, very simple, but it's complicated at the same time. So we can do, first of all, the sequence analysis. We do the gene synthesis. We are using an artificial intelligence database to be able to better code and optimize. That means to have a better yield in performance for the proteins. So what we do afterward, of course, is optimizing the production procedure and then going to the protein purification. And what we can do afterward, just to accelerate your research, we can also do this BLI. So for those who are not very familiar with the BLI, is the biolayer interformity screening. It means we do have, for example, protein or antibody, and we would like to kind of assess which protein will bind better with my antigen. So this is also important. We can also do like 600 samples per day. So just also an overview again. It's also I would say the trend, which is now in Europe, but also even in Africa, a lot of people working on nanobodies, so the VHH or the CVF. We can do 1 milliliter, but we can go till 5 milliliters for 5 business days only. And at the end, you are getting your protein for evaluation within I would say it's a really very short time. So just to give you a bit of an overview, I just put some screenshots. Of course, it will help you to screen, but also to optimize whatever. So here, for example, it's for the protein biomarkers. Alternatively, for the methylated cell-free DNA detects. It's for the early detection for the stage of pancreatic cancer. So if you are not, again, it's about rapidity. If you are not rapid with the rhythm of bioinformatics, we cannot trigger off the right benefit out of it. So, of course, just to have an overview, a summary of the cell-free expression system versus the AI, could be startup or any lab, we are gaining a lot in terms of time. So we don't need to wait 5 weeks, for example, to get the protein, but now only 5 days, small amount, and then we can nurture the bioinformatics tools and also scale up even the cell-free expression if you want to. So in this case, you can analyze the proteins, the hydrophobicity, for example, the linking with the other small biomolecules, but also even the therapeutic target at the end. So just also to show you now, apart from the cell-free application or expression system, we do have this TurboCho expression platform. So this is also an optimized platform that I would say probably some of you are already using it, because I've seen probably some countries, they are getting this TurboCho expression platform. This is also an optimized platform to get the protein in CHO, so expressed in CHO system within 1 week, could be also 2 weeks for very big amounts. So of course, what challenge you are facing? First of all, the budget, how to improve the quality. Sometimes the antibody is not having a good quality, the yield of the antibody, and sufficient capacity, and also the project deadline, because we don't have enough time. With the TurboCho platform, which is also new, just also to update some the innovation that we are having now, because it's as fast as 5 business days, but this time is produced in TurboCho, okay, so in CHO cells. So we are doing the gene synthesis, the plasmid preparation, the transfection and expression, antibody purification, plus the quality control, so that you would be sure that your sequence are validated. So it depends on which stage your project is. It could be small amount, but could be also going even to 200 milliliters, 200 liters, sorry. I would not go too much into details, but just to have an idea about how efficient the platform of TurboCho in terms of yield. We can go only for 1 milliliter, and we can get from 0.1 milligram to 5 milligrams of antibodies. So here, of course, we are speaking about the early stage R&D to lead screening. I mean, you can go also in terms of other details, but just for you to know that this platform does exist. Probably some of you is asking about what kind of format of antibodies that we can do. So TurboCHO can take a lot of antibody formats. If there is no, if it's not mentioned here, we can also discuss, because at the end, GenScript is making a lot of customized projects. Okay, so we can do bispecific, we can do IgGs, we can do whatever format you would like to. It's important for us to get the sequence first, and then we get the sequence evaluated, and then we can discuss further which, for example, volume we could go to have the better yield that you are looking for. So I think just also to give you a bit of, I just put a case study here for the artificial intelligence-driven codon optimization, which is the GenSmart ZQ for GenScript. You can see here. So of course, so we do have the gray GenSmart and the green one, GenSmart ZQ, and then difficult antibodies. Here is milligrams, so we can have a look on the yield. You can see that with in the green part optimized with the GenSmart, we do have a way better yield in producing those kind of antibodies. Same thing, sorry. So same thing for the coming downstream application. So once we do have our antibody produced or our protein produced, we do have a bunch of downstream assays. So whatever you would like to analyze, the general properties, the identification, characterization, the function. So we can do antibody-antigen binding affinity, protein-protein, small molecular interaction, the impurity, customized assay, sequencing, and so on and so forth. So, and all those, of course, downstream assays, we cannot do them, sorry for that, without bioinformatics. So that's very important for all of us to know. So here I just also put a small case study regarding the TurboCHO case. The format here is a human. H kappa, which you have, for example, for only one step of purification, the yield was 374 milligrams per liter. And the purity for the SDA space, for example, was more than 95%, and SEC HPLC more than 99%. So we are not speaking only about the yield, but we are also speaking about the purity of the antibody that you will get. I mean, same thing for the for the other format, for example, the FAB, the SCPF. I would say that more and more we are having more, let's say, trend to go for nanobodies in this period, I would say, those years. And the same thing, the yield is, for example, here we can get 65 milligrams per liter with a very nice purity also percentage. So this is my last part. Apart from the cell-free expression system, the TurboCHO expression system, what I would like to show you here is that apart from that, GenScript is also offering a bunch of free bioinformatics tools that makes your life easier, and it's for free. So let me, I'm not sure if I can just show you quickly. When you go to the website over here, of GenScript, there is a bunch of bioinformatic tools which can really help you. Whatever, if you want to order, for example, from GenScript or not, but you can use it, it's fine. So, for example, if you are working on molecular biology, CRISPR, peptide, protein, antibody, you will find several tools that you can use. For example, codon optimization, codon frequency stable, GenSmart target enrichment, and so on and so forth. Priber design, SI design, you can just click and then use it. If there is, I'm sorry. I'm getting sick with this horrible cold in. And then for CRISPR, I think I had a discussion with some people also in Institut Pasteur Tunis about this tool for the CRISPR. We can also go for being supported by this bioinformatic tool to better design the guided RNA or also the protein itself. So for peptides, same thing, common proteins, antibodies, just go and have a look. So I've just opened two. For example, this peptide library tool. What you will be able to do is, of course, peptide could be several. It could be overlapping peptide library, could be alanine scanning library, whatever. You just need to go here to input your sequences, peptide length, amino acid overlap, and then click, and this tool will help you to design this peptide. Same thing for guided RNA. HDR template tool, same thing if you are working in a knockout or HDR knock-in protocol. Same thing, you will be guided. So which kind of nuclease would you be working on, the target species, the number of guided RNA, and so on and so forth. So this is just a summary of what we can offer and how we can support people who are, familiar with bioinformatics or not, but be sure that people behind bioinformatic tools are trying to simplify this for you. So I think that was it from my side. I would like to thank you. I was quick, but I hope that I was simple and clear enough to pass my message. If you have any question, feel free. But Dr. Hamasi, if you can unmute yourself if there is anything so that I can hear you at least from your side. I'm not sure if I'm able to hear you. The part of Institut Pasteur Tunis is muted.
Siri, can you hear us?
I can hear you now.
Yes, okay.
Okay.
Thanks Siri, for this presentation. Very interactive. So I have two questions. Regarding your here are just specifically, do you have a representative in Morocco?
If there is anyone who can just repeat the question because the voice is not very clear at all. I'm sorry for that.
Okay, okay.
Okay, thank you very much.
Oh, amazing. Thank you so much. Yes, I can hear you. Thank you.
She's hearing that.
Yes, okay. So my first question is if JavaScript have a representative in Morocco or in which country in Africa?
Yes, we do. That's a very good question. I am already dealing with several pharma companies in Morocco, but also in Masir, in U6 MPC. So we do have already a direct market with Morocco, actually. So we don't have any representative, but honestly, I prefer to go direct. So if there is anything you want me to support, feel free to either discuss with Dr. Osama Hamasi or I can also share now my screen in order to get my emails. But for sure, we do have already a lot of business happening in Morocco.
For this information, sure we will need some articles from your company, mainly in terms of proteins and genomes and antibodies. So I'm Fouzia Douani from Institut Pasteur, and I think you can have lots of representative. I know that you have given me the information that you are dealing with Masir and with the UM6P. It's very interesting. Maybe you can deal with the Institut Pasteur of Morocco as well. And about the products that you are providing, are they personalized for each command, or we can have everything in your platform? Thank you.
Yes, that's a very good point. Thanks a lot for this question. Actually, the way we are dealing is we are getting project by project. That means that, especially in my team, honestly, we are taking super customizable projects. So whatever project you have, what we can do is we have 15 minutes discussion through Teams, for example, to explain us your project. Could be, I don't know, protein expression, antibody expression. We can discuss what kind of details we need to evaluate this project, like sequences, the amount that you would like to get, for example, and then we can tell you, yes, we can do or not, or if there is any challenge, it could be always a communication between all of us, actually, to have this project done successfully. So the answer is yes, we are taking a lot of project customizable, and this is actually our core business.
Okay, so hi Serene, I'm Qais. Thank you very much for your contribution and for your excellent presentation, giving us an overview about the GeneScript product. So thank you again, Serene, for this nice, very nice talk, and we will come back to you very soon. Thank you very much.
Thank you so much. Then enjoy your the rest of the day. Yeah?
Now we have 15 minutes break.
Je peux me présenter pas.
Fazer apresentação.
اوكي ثانك يو جمهورنا باش نبقوا
Hi everyone, can you hear me? Can you hear me? Okay. Should I start or? Is my presentation visible now? Okay. So, welcome everyone to this presentation. I am Shoaib. I am a PhD student at RWTH Uniklinik Aachen. I did my MSc in Computer Science from Zarling University and my BSc in Bioinformatics from Comsec University, Pakistan. The work that I'm going to present is actually from before I started my PhD. It was in collaboration with Paulo Martel, who works at the Center for Biomedical Research at University of Algarve in Portugal. So before I start my presentation, which will be on using positional embedding and transformer-based CMA's neural network to predict transcriptome homologs for future protein structure prediction in applications such as genome editing and drug design, I would like to thank Professor Kais and Professor Osama for giving me the opportunity to present, and also Yasmin Othmani, who was here with us in lab for a few months and from whom I got to know about this workshop. So yeah, thank you to all of you. So this work that I'm going to present, our framework, it has two important applications, one in guided RNA design and another one in improved drug design. So our framework, it can help design guided RNAs that can specifically target unique regions in the DNA or RNA. And it can also help identify potential off-target regions that share similarity with the intended target. So if you have, for example, gene A who has, let's say, 90% similarity with gene B, but gene B, let's say it's a paralogous sequence or paralogous gene, and gene A is your desired or homologous gene that you want to for example, cut in your DNA. So our framework, it can help you identify such gene A's and also help identify gene B's that would help you to actually prevent cutting your DNAs at an unintended or undesired locations. And it can also help you identify homologs of previously known drug targets in new contexts or organisms, and it can facilitate drug repurposing by linking existing drugs to new diseases based on conserved homologous regions. So I will just briefly go through a bit of related work. For example, in protein structure prediction, we have traditional methods like X-ray crystallography or NMR spectroscopy, which can help in protein structure determination. However, they're expensive, time-consuming, and often limited to certain protein classes. Recently, there have been a lot of work that has been done in deep learning-based methods for protein structure prediction. One notable tool is AlphaFold, which came out in 2019, which integrates multiple sequence alignment information and a novel representation of protein structures. And then more recently, there is Rosetta Fold, which also combines multiple neural network modules to refine the structure prediction. In terms of homolog detection, we have BLAST, which is basic local alignment search tool. We have SciBLAST, which is position-specific iterative basic local alignment search tool. We have profile hidden Markov models, and again, we have machine learning-based approaches which can do homolog detection, automatic homolog detection. So here is the data set that we used for our study. The data set we used is Hugo Gene Nomenclature Committee data set, which organizes genes into families. So we give the messenger RNA sequences of genes as input to our model. And if both the sequences in a pair, they belong to the same family, the pair is considered as a homolog and vice versa. So the training data set, it consisted of around 17,160 pairs, whereas the validation and test data set consisted of around 5,700 pairs respectively. So here you see if we have two sequences, two input sequences, and if both of them, they belong to the same family, we term that input pair as homolog, and we label it by 1. And if the input sequences, they are from different families, we label them with 0, which stands for non-homologs. So the first step in our methodology is to create one-hot encoded vectors from our sequence data. And from those one-hot encoded vectors, we create positional embeddings. So every nucleotide in the sequence, it is represented by a 4x4 array. So for example, this 4x4 array, it represents a single nucleotide in the sequence. So this basically captures the positional information as to how much of which nucleotide each position corresponds to. So here, every nucleotide is represented by a fraction of A, a fraction of U, a fraction of C, and a fraction of G. And here is the overall architecture of our unsupervised CMS neural network. So we give our one-hot encoded data as input. So this goes through a token encoding and a position encoding module, which is then added together to create a representation that goes as an input to our transformer model. So first of all, here we have a self-attention network. It is actually a multi-head self-attention network, which helps to capture the positional information in the sequence data. And then, so our data is basically actually decomposed into 3 vectors, Q, V, and W. Sorry, Q, K, and V. And then after that, the representation that we get, we apply a dropout and normalization layer, followed by an activation layer, followed by a linear layer, again a dropout layer, a normalization layer. So these dropout and normalization layers, they are actually added to avoid overfitting of the model. And then the representation from both the sequences, so the same network is being run on both the input sequences. And this transformer block, it actually shares the weights between both these runs of the network. So at the end, with the representation that we get, we can concatenate that representation. Again, we apply a dropout layer, followed by an activation layer, followed by a dropout layer. And in the end, we get an output embedding. So the output of this network is actually a learned representation of the input pair, which is passed then through a custom similarity measure, which in our case is the Euclidean distance. And we employed a similarity loss during training in order to encourage the network to assign higher similarity scores to the pairs of messenger RNA homologs. So we evaluated our model using a few evaluation metrics. So we employed reconstruction loss. So we have actually employed mean squared error as a reconstruction loss to assess the quality of the learned embeddings. So this mean squared error, it measures the dissimilarity between the original sequence and the reconstructed counterparts from the embedding. So it is computed by this equation where the mean square error for a particular pair, it is computed as the sum of the representation. It's actually computed as a difference of the input representation with the learned embeddings in the output layer for one of the sequences in the input pair. And the same is being done for the second sequence in the input pair. We add both of them together, we divide it by 2, we take its square root, and we do it actually for all the sequences in our input data, or all the pairs in our input data. We employed average log density estimation, in particular kernel density estimation, to estimate the probability density function of the embeddings, which also helped us to identify the regions of high density and potential outliers in the embedding space. So the average log density, it is computed by this function where we compute the log density over all the newly generated samples, and the log density for a particular sample, it is computed as a log of the Gaussian kernel being applied over the new samples that is being generated by the distribution. We did cluster variance analysis, in particular intra-cluster variance analysis of the embeddings to evaluate the compactness of the clusters which are found by the CMS network, where the lower intra-cluster variance indicates tighter and more distinct clusters. So the mean intra-cluster variance in the end, it is basically simply computed as the average of the variances for each of the clusters. So to do the prediction at transcriptome level for each messenger RNA, we determined its most similar homolog by selecting the nearest embedding in terms of Euclidean distance between the learned representation. So we deem the prediction as correct if the family of both the RNAs, they are the same. We deem the prediction as correct. Otherwise, if the families of both the RNAs, they are different, we deem the prediction as incorrect. In order to do the prediction at protein level, we use SciBlast in order to examine the homolog of protein counterparts of the messenger RNA sequences by cross-referencing predicted homolog, transcriptome homologs with the SciBlast results. And we leverage the SciBlast results to identify if the predicted messenger RNA homolog also shared the homology at the proteome level. So we deem the prediction as correct at the protein level if the protein counterpart of the homolog is among the SciBlast hits, and we deem the prediction as incorrect otherwise. So we employed gradient accumulation to enhance the efficiency of the training process because of the high dimensionality of the data. So the overall procedure, it was organized into accumulation groups where each group spanned k consecutive iterations over the training mini batch. And for each iteration within an accumulation group, we computed the individual batch loss for each batch. And after we are done with the accumulation group, we applied the accumulated gradients to update the model's trainable parameters. So in our transcriptome similarity dictionary, we got 877 pairs in total, and the similarity between the pair of input to messenger RNA sequences is actually computed using L2 normalization of the Euclidean distance of the learned representation. So for each input pair, we get an L by M embedding matrix in the output layer. And the Euclidean distance for each nucleotide position k is calculated as taking the value at the 0th sequence position for a particular nucleotide k and subtracting it by the sum of all the nucleotide values that you get for that particular nucleotide position. So once we have the distance for each nucleotide position, we can actually calculate the L2 norm of the resulting distance vector. You can actually calculate the L2 norm of the resulting distance vector, which would in turn give us a single value, and that single value can be used to calculate a similarity score given by 1 over 1 plus the L2 norm that was calculated here. So we got an impressive 78.79% of correct predictions at the transcriptome level. When we cross-reference with the SciBlast results, we achieved a modest score of 41.72% correct predictions. We got an average log density score of minus 4.53 into 10 to the power 4, which signifies the model's ability to generate samples consistent with the underlying data distribution. We got a mean intra-cluster variance of 0.56 into 10 raised power minus 20, which showcases that our embeddings, they effectively grouped similar messenger RNA sequences together. We got a reconstruction loss of 0.17 into 10 raised power minus 8, which is again a very low value, which indicates a better reconstruction performance. And to conclude this presentation, we basically worked on developing a novel approach that combines positional embeddings with a transformer-based CMS neural network to predict transcriptome homologs and the accurate transcriptome homolog identification. It can enhance the protein structure prediction as well. And our framework addresses critical challenges in genome editing, drug discovery, and protein structure prediction by leveraging the power of AI machine learning based bioinformatics, AI machine learning driven bioinformatics. And the ability to identify homologous sequence supports genome editing by enabling precise guide RNA design, minimizing off-target effects, and targeting the conserved regions for effective genetic interventions. Our approach, it also streamlines target identification and aids in drug binding site recognition by leveraging shared structural and functional features among homologous sequences. So here is the list of references that I've used for this presentation. Thank you. I hope that I was brief enough and I finished my presentation in time. If there are any questions or any feedback, you guys are most welcome. Can you guys hear me?